today we are going to talk about microbial analysis of milk and milk products. In microbial analysis of milk and milk products, we are interested with two aspects. One is the enumeration of microorganisms. Enumeration of microorganisms refers to establishment of the number of microorganisms present in milk and milk samples. High number of microorganisms means that the milk is going to be degraded very fast and therefore will have a lower shelf life. The other aspect is isolation of microorganisms. Isolation of microorganisms is the establishment of the type of microorganisms present in milk and milk products. Some microorganisms are associated with diseases and therefore will render the products unfit for human consumption if they are present in milk and milk products. In carrying out microbial analysis, we need to carry out some preparations. The first preparation is preparation of the glassware. Preparation of the glassware involves cleaning of the glassware, rinsing them, drying them, and sterilizing them. Sterilization of glassware is by use of hot air oven, which is set at 170 degrees for a period of 1.5 hours to 2 hours, and then you cool the glassware to 47 degrees centigrade. All the glassware should be locked during sterilization. Pipettes are put in a pipette canister. After glassware are sterilized and ready for use, then you prepare the media. The media preparation is usually by following manufacturer's instructions. However, sometimes you may need to prepare more media or less media than indicated by the manufacturer, and therefore you need to carry out a calculation before you can weigh the amount of media required, and then you disperse it in the distilled water, and then most often it is sterilized by autoclaving at 121 degrees centigrade or 15 psi for 15 minutes, and then you cool the media to 47 degrees centigrade. After you are through with the media preparation, then you prepare the dilution. Dilution making is through mixing milk sample with diluents already made. The purpose of dilution making is to thin out microorganisms. Diluents are normally prepared as per manufacturer's instructions, and then 9 ml of the diluent is distributed in several test tubes. To make dilution 1, you transfer 1 ml of milk sample to first tube with 9 ml of diluent. This will make dilution 1/10. To make dilution 2 or 1/100, you transfer 1 ml of dilution 1 to the second diluent. And you continue like this, what we call serial decimal dilution, until the required number of dilutions are made. After that, the second step is inoculation. Inoculation is the transfer of dilutions and mixing them with the media so that the microorganisms can start growing. When you carry out inoculation, you should ensure that there is no contamination. We follow what we call aseptic techniques during this process of inoculation. After inoculation, the next step is to incubate. Incubation is placing your samples in the right environment for growth of microorganisms, and this is in terms of temperature, humidity, and also time. After the expiry of the incubation period, then you come back and establish the number of microorganisms present. This can be done by counting of the colony forming units if the test is on a solid media, or by estimation of the number of microorganisms if the test is being done on a liquid media. And then you use tables to estimate the number of microorganisms present. This is how you carry out microbial analysis of milk and milk products.